Hello, welcome to Flip. So today we'll be learning how to identify gases. First of all, we start with ammonia, which is NH3. What we do is we place the damp litmus paper near the mouth of the test tube, like so. And you can see the red litmus paper will turn blue. Okay, so this is our red litmus paper, and it will turn blue like this. Okay, this is because ammonia is alkaline in nature. Okay, next we move on to carbon dioxide, which is CO2. How we identify carbon dioxide is when we bubble gas through the test tube containing lime water, which is CaOH2, which is calcium hydroxide. So we can see that the gas will move through the tube here into the colorless CaOH2. Okay, what will happen next is that this thing will turn white, okay, inside the test tube, and that is when we call it to be chalky, and that is the white precipitate that's formed inside the test tube. Okay, you can see that colorless, now it turns to white. Next is chlorine, which is how to identify chlorine. It's actually the opposite of ammonia. What we do is we place damp litmus paper near the mouth of the test tube, but in this case we place a blue one. Okay, and we can see that the blue litmus paper turns red after a while when it's in contact with the chlorine gas, and if you put it longer, okay, it will actually turn white, and it's because it's bleached after a while. That is how toxic a chlorine gas is. Next, we will learn about hydrogen. So hydrogen, how to identify hydrogen is just to place lighted splint inside the test tube. So imagine I have this test tube contains hydrogen. Okay, lighted test tube means the test tube is on fire. So when I put this tube inside this test tube, this lighted splint will be extinguished. Okay, and it will emit this pop sound. Oxygen is very similar. It's just that the splint now, instead of being lighted, is just glowing. Glowing just means it's lighted a bit on the tip of the splint, something like a joystick. So when you put this glowing splint inside the test tube containing oxygen, what happens is that this splint, okay, when it's for example, it's orange in color. Now when you put it inside, actually it glows brighter to maybe say red color. Okay, so rekindle, the word rekindle really just means to grow brighter. Okay, and that's the difference between hydrogen and oxygen identification. One is lighted splint, one is glowing splint. Finally is about sulfur dioxide, which is there are two ways to identify sulfur dioxide. First one is we dip the filter paper, okay, this one is our filter paper for example, into acidified potassium manganate 7. Okay, and originally a white color filter paper will turn purple because potassium manganate is purple. And then we place this filter paper at the mouth of the test tube, like what we have done in the case of ammonia and chlorine. Okay, we just put it at the mouth of the test tube, and we will see that this filter paper turns from purple, okay, into white color. Okay, or another way is that we can bubble the gas, like what we have done in the case of carbon dioxide. Okay, bubbling. We bubble the sulfur dioxide gas through this tube here into the test tube directly, and this potassium manganate, which is originally purple in color, will now turn colorless. Okay, it will just turn transparent to a transparent liquid. And that is all for the identification of gases.